Hi guys, welcome to Master IB.
So today we're going to be covering some basic biology in which we're going to cover prokaryotic and eukaryotic cells. So let's get into it. Prokaryotic cells are very simple in structure. They are even very small. They're less than 10 micrometers. A classic example of a prokaryotic cell would be E. coli, which is a type of bacteria. In terms of the structure of a prokaryote and its organelles, it has a cell wall, cell membrane, cytoplasm, a nucleoid where its prokaryotic DNA is stored, has a plasmid where additional DNA is also stored but is not consistent in state, so it can either be removed or re-replicated. It has 70S ribosomes, which is used for polypeptide synthesis, and unlike the eukaryotic cell, they have 80S ribosomes, so that's the difference. They also have flagella, which is used to help with the locomotive of the cell, which means it helps it to move around. It has pili, which is hair-like extensions found within the bacteria. So what's special about the prokaryotic cells is that they don't actually go through sexual reproduction, unlike the eukaryotic cells. They would be going through sexual reproduction, whilst prokaryotics go through asexual reproduction. Now this happens through the process of binary fission, which is basically the cell's DNA replicating semi-conservatively.
Now to eukaryotic cells. Eukaryotic cells are bigger than 10 micrometers as opposed to the prokaryotic cells, which are smaller than 10 micrometers. Another difference between the two is the fact that eukaryotic cells are compartmentalized, meaning that it has organelles which are separated from each other by a membrane. One example of one of these organelles is the nucleus. The nucleus has a double membrane with pores in it. The nucleus function is to contain the DNA. In the eukaryotic cell, the DNA is contained within this nucleus and is associated with histone proteins. Contained within this nucleus is the nucleolus. The nucleolus makes ribosomes, which can either be free ribosomes or ribosomes bound to the surface of the rough endoplasmic reticulum. The free ribosomes make proteins, which are used within the cell. So those can be enzymes used within the cell or any other kind of protein that the cell needs. While the proteins made by ribosomes on the rough endoplasmic reticulum are proteins which are destined to go fulfill a function outside of the cell. This happens by the ribosome producing the protein entering the cisternae of the rough endoplasmic reticulum. As previously mentioned, there is the Golgi apparatus, where the protein is sent to when it leaves the rough endoplasmic reticulum in a vesicle. So the protein arrives in the cis section, which is CIS, of the Golgi apparatus, is then modified and leaves the Golgi apparatus to the trans section to be secreted out of the cell. Another example of an organelle is the mitochondrion. The mitochondrion also has a double membrane and is the site of many processes of aerobic respiration. Some organelles are only present in plant cells and some are only present in animal cells. For example, chloroplasts are organelles which are responsible for the light-dependent reactions of photosynthesis. There's also the smooth endoplasmic reticulum, which has the function of producing lipids. So as previously mentioned, after the protein leaves the rough endoplasmic reticulum in a vesicle, it enters the Golgi apparatus. To be specific, it enters the cis section of the Golgi apparatus, and the protein leaves through the trans section. In the process, it gets modified for the use that it is destined. So there are some organelles which are only present in animal cells and some organelles which are only present in plant cells. For example, the chloroplast is an organelle with a double membrane which is responsible for the light-dependent reactions of photosynthesis and is only present in plant cells. It has a double membrane, as previously mentioned. Inside the center, it has stacks of thylakoids which contain the pigment chlorophyll, which absorbs the sunlight. Those are stacked in stacks called granums, and the granums are connected by intergranular lamella. Now the cell wall is also an aspect that is only present in plant cells, but it cannot be described as an organelle as it is in the extracellular matrix, which is because it is outside of the cell membrane. The cell wall gives the plant cell shape, similar to the cell wall in the prokaryotic cell. However, as opposed to the cell wall in the prokaryotic cell, it is made out of cellulose, hemicellulose, and pectin. Now, organelles which are only present in animal cells are things called lysosomes. Lysosomes are basically vesicles filled with digestive enzymes, which are used to digest food that the animal cell takes in. Now, the fact that these digestive enzymes are contained actually saves the cell, because the digestive enzymes actually have the power to eat the cell itself, which shows an advantage of compartmentalization. Other things that only animal cells can have are flagellum, which is the same kind of flagellum as in the prokaryotic cell, and cilia, which are basically projections on the outside of the cell, which can either move a cell through a liquid or can move a liquid around a stationary cell. Now lastly, the difference between prokaryotes and eukaryotes is that prokaryotes divide by binary fission, as previously mentioned, but eukaryotes divide by mitosis, asexually, or by meiosis in order to produce gametes, which are then used for sexual reproduction. A difference between the prokaryotes and the eukaryotes is that opposed to the prokaryotes, the eukaryotic cells have ATS ribosomes, which are slightly larger. So that's it, guys. Now to test yourself, here's an actual IB question. What do prokaryotic cells have that eukaryotic cells do not? A. Mitochondria, B. 70S ribosomes, C. Histones, and D. Internal membranes.
Alright guys, please like and subscribe. That was our video. Hope you enjoyed it. And leave the answer down below. Thanks.